Next, we're going to talk about T lymphocytes, better known as T cells. We're going to talk about the specificity of the T cell receptor, or TCR, and the types of T lymphocytes. The TCR, or T cell receptor, is like the BCR in specificity, but it's not shaped like the BCR. It's not shaped like an antibody, which kind of makes sense since T cells do not make antibodies. We have a cytoplasmic membrane, that membrane portion, which embeds the TCR in the cell membrane. We have a constant region, and like with the BCR, we have a variable region. Like with the B cells, we get these by gene shuffling, so that the antigen binding site is different for each T cell. Also, unlike B cells, TCRs are unable to recognize soluble antigens. They recognize antigens that are associated with major histocompatibility complex proteins, or MHCs, which we're going to be talking about a little bit later. There are three basic types of T lymphocytes. Cytotoxic T lymphocytes act like natural killer cells. They go around looking for their specific antigen on cells. Now, unlike the natural killer cells, like I said, they're looking for their specific lymphocytes, so they are a part of the adaptive immune response. Then we also have helper T lymphocytes. They go around helping other T cells and B cells and macrophages. There's the ones that we're going to talk about in this class. There's type 1, which goes through and helps macrophages function better. So they go through and they will attach to macrophages that have swallowed down as many bacteria as they can. They're full of phagosomes, they can't do anymore. And they kind of give the macrophages an antacid. They stimulate them to digest faster and to phagocyze more. So they upregulate macrophages. Type 2 helper T lymphocytes work with B cells to produce more plasma cells and produce more antibodies. That's why we call them helper cells, because they go around talking to the others. Then we have regulatory T lymphocytes that go around calming things down, downregulating the immune response. Now let's talk about development of B cells and T cells. So, hemopoietic cells divide, they form lymphocytes, some of them become B cells, some of them become T cells. Now they need to undergo part of the maturation process called clonal deletion. In other words, we're going to get rid of B cells and T cells that recognize your antigens. We don't want them attacking us. So let's start with T cells because it's more complicated. So here we've got a stem cell. Here we've got a whole slew of T cells that are all ready to go on to their next step. Then they go to the thymus, and there are specialized thymus cells that within them they produce all of the proteins that are made in the body, even if they're not normally made by thymus cells. Things like hemoglobin. Hemoglobin usually is only made by red blood cells, but you've got to make sure that your T cells won't attack red blood cells. That would be very bad. And they display these proteins on their major histocompatibility 1 proteins. Now remember that T cells only recognize antigens that are bound to MHCs. So the first test is, do we recognize this particular body's MHC1? If the answer is no, they're stimulated to undergo apoptosis and destroy themselves. If they do, then they receive a survival signal. Then if they recognize an autoantigen, then they can stay alive and become further along the path of becoming T cells. If they do recognize autoantigens, most of them undergo apoptosis. Some of them become regulatory T cells. B cells, on the other hand, don't go to the thymus, but they undergo a very similar process. They're exposed to autoantigens during their development. If they recognize autoantigens, they either undergo apoptosis or they're kind of put to sleep, or they can change their B cell receptor. In other words, they go back and they say, oh, well, I can fix that. If they don't recognize autoantigens, then they go on to the spleen to further development, and then they go on to the lymph nodes. Next up, immune response cytokines. Immune cells talk to each other, and the chemicals that they use to talk to each other we call cytokines. So let's go through the 5 classes of cytokines. Interleukins, as the name implies, are released by leukocytes as signals among leukocytes. Now, a few other cells do release interleukins, but the vast majority come from leukocytes. Interferons, we've talked about. Interferons, they are antiviral proteins, and they are mainly released by virally infected cells, but interferon gamma is a phagocyte activator. So it's released by cells that are infected, they're released by leukocytes, and they tell the phagocytes to come on over here, we got a problem. Growth factors stimulate leukocyte stem cells. There are other growth factors that stimulate other cells to develop, but of course in this chapter we're mainly interested in getting more leukocytes. Tumor necrosis factor is released by macrophages and T cells. It kills tumor cells, that's where we originally got the name from, but it also regulates the immune response and the inflammation response. Now the last one, chemokines, are involved in chemotaxis. They're released by leukocytes to tell other leukocytes to come on over, this is where the fight is. Well, that's it for this topic. It was quite a big topic, and here are reminders of the learning objectives for this topic.